Hi, Satya. You have to switch your microphone on.
Thank you very much, Catalina, for your nice presentation. I think we'll come back to you after all the talk finished for the question session. So I think my next objective is to introduce our all four presenters. So the first one will be Dr. Cecilia Caldavila. She's working as an investigator at Biogenesis Bago, Argentina. And many of us know Biogenesis Bago and their role controlling FMD in South America through providing efficacious vaccines along with other established manufacturers. So Cecilia is presenting an interesting and a timely topic. As we know, since 2009-2010 outbreaks in Southeast Asia and Far East, there is continuing issues on the performance of some of the existing vaccines. And her title of talk is FMD vaccines containing O1 campus A24 Cruzerio and A2001 Argentina, high antigenic relatedness and cross protection upon challenge with Asia 1 97 and O Middle East South Asia India 2001 E lineages. Our second speaker is Dr. David Mackay, and many of us from UFMD currently, and many of us know Dr. Mackay. And many years he was a FMD researcher at Fulbright Institute, then led Fulbright Institute Epidemiology Division as a head, and then worked several years in EMA as a regulator, and recently working in UFMD at FAO. I happen to be a colleague to him both in Fulbright Institute and currently at FAO. He is presenting on an important topic, the role of testing in pre-qualification of vaccines against foot and mouth disease. Our third speaker is Dr. Julian Sego, and Dr. Sego is working as a group leader at the Fulbright Institute on FMD and classical swine fever. His talk on stable FMD vaccine for East Africa is very practical and may solve an important issue in endemic countries where very high ambient temperature exists. And our fourth selected talk is will be delivered by Dr. Jeff Hammond. And I know Dr. Jeff Hammond as past WRL FMD head at Fulbright Institute and recently working as FMD industry expert at GALMED. The project he will describe us may bring a fruitful control of FMD in East Africa and may be example for other endemic countries. Thank you very much.
Good morning to all of you. I'm Cecilia Caldevilla. I'm working in Biogenesis Bago, Argentina in FMD Department of Development and Research. I'm going to present this work titled Foot and Mouth Disease Vaccines Containing O1 Campus, A24 Cruzeiro, and A2001, High Antigenic Relatedness and Cross Protection Upon Challenge with Southeast Asian and G7 and in 2001 lineages. This work was performed in collaboration with SENASA, OIE FMD Reference Laboratory in Argentina. As a brief introduction, foot and mouth disease is the most economic, devastating disease worldwide. It produces a decrease in productivity of livestock industry. The causal agent is the FMD viruses, is constantly evolving and changing, and this represents a challenge for the properly selection of vaccine strain that could confer broad protection. Foot and mouth disease is endemic in many parts of the world, particularly here we are going to talk of the Southeast Asia region. It has a complex epidemiological situation. There is a co-circulation of multiple serotypes and lineages. Here you can see the CR region in this map and the relative prevalence of the different serotype O and A that are causing of the current outbreaks in the region. The in vitro vaccine matching is an essential tool for vaccinate control policy, and it's based in the determination of the R1 values. Nowadays, there is a lot of reports that mention the limitation of the R1 values because exists the possibility of that high potency vaccine will even have R1 value lower could confer protection. So nowadays it introduced the concept of the high heterologous neutralization titers, considered also as a good indicator of protection. South American vaccine strains are well-established vaccine strains used in systematic vaccination in South America and formulated in high quality and high potency vaccine strains have demonstrated a broad antigenic coverage and cross protection against heterologous Asian viruses. The principal objective of this study was to assess in vitro vaccine matching of these South American vaccine strains included in high-quality vaccines against heterologous viruses prevalent in Southeast Asia, and also to assess the efficacy of the potency by challenge with prevalent lineages in 2001 and Southeast Asia 97. First, we assess the in vitro vaccine matching. For this purpose, 2 serum pools of swine were prepared from 5 serum each with medium to high titers. It was included swine vaccinated with monovalent O1 campus high potency vaccinated at 21 dPV and swine vaccinated with trivalent high potency vaccine at 28 dPV. The vaccine matching was assessed by the bidimensional virus neutralization test to infer cross protection. The R1 value is a coefficient that represents the match of the vaccine strains against the field isolates. And this is the formula for the calculation. It is considered a match value above 0.3 that the vaccine could confer protection, and meanwhile if it is below 0.3 it considers that the vaccine was not able to confer protection. But as I mentioned before, now the R1 value have several limitations, and it's important to consider the high heterologous titer as an indicator of cross protection. In this cross-reactivity assays, the file isolates were provided by Senasa, included all these viruses belonging to serotypes O with the representative topotypes on lineages of the region, and the viruses for the serotype A. Then we assess the in vivo challenge test, and the trial test was performed in accordance to the European Pharmacopoeia in biosecurity for OIE level. The challenge viruses were provided by Senasa, adapted in swine. The commercial vaccines were Viaptogen, trivalent O1 campus A24 and A2001, manufactured by Biogenesis Vago, containing more than 6 pt2 of the homologous vaccine strains, water and oil single emulsion with purified antigens. Here you can see the experimental design. It was performed 4 independent trials for each one of the challenge viruses. In one of the trials, we include 3 groups of 6 pigs vaccinations with reduced volume of vaccines, and the challenge was performed at 28 post-vaccination with 10,000 infectious doses by the intramuscular route in the foot. The animals were observed during 8 days post-challenge. Those showing lesions at sites other than the inoculation site were considered and not protected. The PD50 value conferred by the vaccine were calculated by the Spearman-Carver method. Now I'm going to show you the results of the in vitro vaccine matching against ophil isolates. We analyzed swine sera, post-vaccinal sera, collected at 21 DPV with O1 campus high-potency vaccines. Here you can see for all the isolates tested, the heterologous neutralization titers were around 2 logs. This indicates a good cross-reactivity of the O1 campus vaccine strains, and inconsistent with this heterologous titer, in this case the R1 values were also above 0.3, indicating the O1 campus has a good cross protection. On the other hand, we assess the cross-reactivity against serotype A ophil isolates. In this case, the serum providing from sera pigs vaccinated with trivalent vaccine O1 campus A24 and A2001 collected at 28 DPV. In this table, you can see the heterologous titer and R1 values for the vaccine strain A24 Cruzeiro, and in this table the heterologous titer and the R1 values for the vaccine strain A2001. For all the viruses studied, the neutralizing titer was satisfactory, and also the R1 values. Here the results of the first PD50 test. The challenge viruses was the in 2001, South Korea 2011. This is an schematic representation of the challenge trial. 3 groups of 6 pigs vaccinated with different volume dose. The lecture performed at days for vaccination showed that none of the pigs received a full dose of vaccines showing any clinical lesion, while 1 vaccinated with 0.5 milliliter dose and 5 with a reduced dose demonstrated disseminated disease, and the 2 unvaccinated controls. Also, the PD50 value of the vaccine was 8. And here I summarize in this table the other trials performed against the topotype Asia, Southeast Asia, and South Korean viruses. In all of the cases, the PD50 value of the vaccine were above 6. As a conclusion, the in vitro vaccine matching test based on the determination of R1 value and the heterologous neutralization titers predicted satisfactory cross protection against serotype O1A file isolates lately circulated in Southeast Asia region. The results of the in vitro assays were in line with the ones observed in the in vivo challenge, but Bioactogen confirmed more than 6 PD50 for all the viruses studied. So the data presented indicate that the South American vaccine strains, when are formulated in a high quality and a high potency vaccine, are an effective tool to control FMD in Southeast Asia region. Thanks for your attention, and thanks for the OIE FMD Reference Laboratory in Argentina, SENASA, for the collaboration in this project.
My name's David Mackay, and I'm going to talk about the role of testing in the pre-qualification of vaccines against foot and mouth disease. This is some work that I've been doing with Fabrizio Rosso in the European Commission for the Control of Foot and Mouth Disease. In this presentation, I'm going to describe the pre-qualification scheme for foot and mouth disease vaccine that we're developing. I'm going to talk a little bit about the current testing performed on foot and mouth disease vaccines and some of the current challenges that exist. And then I'm going to talk about the approach that we're developing to testing overall for the pre-qualification scheme. So first of all, what is pre-qualification? Pre-qualification is an independent assurance that a foot and mouth disease vaccine at least complies with the minimum quality standards that are defined in the OIE terrestrial manual. The approach is modelled on the WHO pre-qualification scheme for human vaccines and has been adapted as is required for the veterinary domain. Only vaccines that have been approved by at least one national competent authority are eligible for pre-qualification, and vaccines have to be manufactured in compliance with GMP. As GMP means different things to different people, the documentation for pre-qualification specifies that GMP is defined in Chapter 2.3.3 of the OIE manual. Applications for pre-qualification will be scrutinised by a panel of independent experts and will consist of information on the pharmaceutical quality, the method of manufacture and quality control, information on laboratory and field trials, and on experience of use on the vaccine in the field following formal approval. The intention is that the EU FMD will publish on its website a list of pre-qualified vaccines that will be available to risk managers and to any people who might be seeking to procure vaccines, who will thereby have a source of information on vaccines of assured quality. The intention is that this scheme will be up and running later in next year in 2022, and in the first instance the scheme will rely on documentary review, but at a later stage that we would like to introduce independent testing. And the subject of this presentation is really what testing should be done and by whom. This is quite a complicated slide, and my intention in showing it is really just to demonstrate that there is a lot of testing that is done during the life cycle of a foot and mouth disease vaccine, which is illustrated in the left-hand column here, and that it is done by a number of different parties. In this case, I've illustrated testing done by the manufacturer, by independent reference laboratories, and by official control laboratories for a number of different purposes. And therefore, when introducing new testing, it's very important to be clear at what stage that testing is being done and for what purpose. And for the purposes of pre-qualification, the testing would be done during the post-authorization stage by an independent laboratory and would therefore fit into the testing scheme here. On the next few slides, I've attempted to summarize some of the key challenges that FMD vaccines represent to regulatory or to independent testing as compared to other types of vaccines. Let's have a look first at the question of strain selection. So for most vaccines, the relevance of vaccine strains is studied only once, and that information is then submitted as part of the vaccine authorization dossier. And the information consists of a combination of antigenic and genetic characterization studies. And I'm sure most of the audience is familiar that for FMD, this is particularly challenging due to the wide antigenic diversity between strains, which leads to the need for multiple master seeds in order to provide a sufficiently wide antigenic spectrum within a single serotype of foot and mouth disease. And this in turn leads to a complex relationship between antigenicity, potency, and efficacy for each serotype of foot and mouth disease. And then it's very difficult for independent and official laboratories to obtain, calibrate, and standardize tests and materials independently from manufacturers. In this context, the recent development of standard antigen panels, such as the Eastern African FMDV reference antigen panel by the WRL at the Pirbright Institute, represents a significant step forward, and I'll illustrate this on my next slide. So this slide shows illustrated data from 5 animals vaccinated with a quadrivalent vaccine containing serotypes A, O, SAT1 and SAT2 in a virus neutralization tests against 16 field strains from the 4 serotypes present in the vaccine to examine the ability of post-vaccinal sera to neutralize the field strains. Using standardized antigen panels such as this, it is possible to examine the epidemiological relevance of vaccine strains against a range of field strains likely to be found within the target area for a vaccine, and therefore examine its potential fitness for purpose within a particular region. Moving on now to look at the question of potency and efficacy. Foot and mouth disease is almost unique in that for regulatory purposes it is permitted to study efficacy using the vaccine strain itself. Whilst this is very useful and it certainly allows the manufacturer to demonstrate the efficacy of the vaccine under the most ideal circumstances in terms of antigenic match, it can lead to confusion between demonstration of compliance with the terms of the authorization in terms of demonstrating potency against the vaccine strain with demonstrating fitness for purpose in terms of expected efficacy against field strains. I think also we're aware that in terms of efficacy, the definitive test relies on challenge in cattle, and that in vitro alternative measures of efficacy are accepted, provided that correlation with protection has been shown. Potency, batch potency, efficacy, onset and duration of immunity, and claims, if they are made, for protection against strains other than the vaccine strain are all evaluated as part of the authorization procedure. So after that rather quick and high-level overview of the particular challenges for regulation and testing of foot and mouth disease vaccine, what are the conclusions in terms of pre-qualification? I think the first is that it'll be necessary to review all of the data on genetic and antigenic characterization of master seeds and the antigenic nature of the vaccine in relation to the information provided on the qualitative composition of the product, in other words, the antigenic nature of the product. It'll be necessary to review data on consecutive batch production records to confirm compliance with minimum international standards, particularly in relation to the minimum OIE standards of either 3 or 6 PD50. to look at the consistency of production and compliance with the authorization and the product literature, the labeling and minimum claims, and to look at any information provided on testing of the product by independent reference laboratories. And what about the future? I think it will be very important to work with reference laboratories about how to carry out testing using standard panels of standard reference viruses and anti-sera for the major vaccine strains, and how to develop those panels further in future, and to continue dialogue with stakeholders on how to improve transfer of technology and materials for product testing. And just to bear in mind that this technology of transfer and materials happens already for many other types of vaccines to official laboratories as part of authorization procedures in both the US and the EU. Thank you very much for your attention.